Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So we were learning about stages of X chromosome inactivation in mammals. So we are learning the step-by-step procedure which is followed in mammals for doing the X chromosome inactivation. So this X chromosome inactivation does the dosage compensation in mammals. So we learned about the first two stages, the first being counting part. So in this counting part, we determine the X chromosome to autosome ratio, which helps us to make the decision whether the X chromosome inactivation is needed or not. Then the second step is choice, like the choice of which X chromosome is going to be inactivated. So that could be either random or it could be skewed. So in the counting part, we learned about the X-linked factors such as RNF12, how the RNF12, which was discovered in 2009, how it helps us to determine whether the X chromosome inactivation is needed or not. So first you establish that there is need of X chromosome inactivation, and after that decision, you have to decide whether the X inactivation would be random or it would be imprinted. So depending on that, we initiate the X chromosome inactivation. So we learned about the first two stages. First, we do the counting, which helps us to determine about the X chromosome inactivation. So in the counting part, once we determine the ratio, it is found out that yeah, you need the X chromosome inactivation. Then you move further to the choice of which X chromosome is going to be inactivated. It could be random, it could be skewed. In that lecture, we learned about Rett syndrome, how the skewed choice can result into some disorder. The Rett syndrome is a neurodevelopmental syndrome. So we learned about that in that lecture. Then we learned about the X chromosome inactivation center, the XIC. How that XIC, which is a region in X chromosome, which is important in X chromosome inactivation. So today we are going to learn about initiation of X chromosome inactivation, the third stage of X chromosome inactivation. So we know that the XIST long non-coding RNA is responsible for X chromosome inactivation. When we were learning about XIC, we learned that the XIST, it has all of the parts, all of the regions are non-coding except the RNF12. So the main regulatory element is the XIST long non-coding RNA. It is going to do the X chromosome inactivation. Today we are going to learn about the mechanism by which XIST establishes the heterochromatinization in X chromosome. So we learned about XIST long non-coding RNA, which does the inactivation of X chromosome. Okay. We also learned about the antisense transcript of XIST, which is known as T6. Okay, which has antagonistic role compared to the XIST. Okay, and we also learned about other regulatory elements such as FTX, JPX, DXPAS 34. Okay, then TSX, Excite. Okay, we also learn about all those other regulatory elements. So today we are going to learn about the mechanism by which XIST long non-coding RNA, it initiates and then it establishes X chromosome inactivation. So the first step is counting, then choice, then initiation, and then there is establishment of X chromosome inactivation. Once it is established, it spreads on all over the X chromosome, and then comes the maintenance. The X chromosome, which is inactivated, it is maintained in that cell throughout its lifespan and then in its progeny as well. So we are going to learn about the further stages of the X chromosome inactivation. So let's first learn about XIST long non-coding RNA. So XIST, this long non-coding RNA, this works in cis fashion. Okay, I hope you know about cis and trans manner of working of regulatory elements. If you don't, I'll explain it in brief. So let's assume that regulatory element. Let's take the example of operator. This is our regulatory element for the gene expression and repressor as well. So we know that operator is a sequence. Okay, so this is operator sequence. upstream to the promoter sequence. So this is promoter, this is operator. Operator sequence could be within the promoter as well, but I'm taking the operator sequence upstream here. So we know that on the operator sequence, the repressor binds. Okay, and once the repressor binds to the operator sequence, if the gene could not be transcribed, so it inhibits the gene expression. Okay. So this is how operator and repressor, they play the regulating role, the regulatory part, like they do the gene regulation. So the operator works in cis fashion. Okay, so like if the regulatory element, it works from in the on the origin of the DNA. Like operator is a sequence, it is a DNA sequence, right? So it works only on the DNA of the origin, right? While the repressor, it can be transcribed from the gene which is maybe upstream here. So this is the gene which codes for repressor protein, but this repressor protein can work in trans fashion, means it can work on the DNA which is not of its origin. So like it can work on this DNA as well as it can work on other DNA strand. So it can bind to the other operator sequence which is present on the different DNA strand. This is the repressor binding to the operator sequence which is present on the other DNA molecule. Let me explain it in a different manner. So for example, there's a long non-coding RNA. Let's name it XYZ. Okay, this is a long non-coding RNA. Okay, and this is the DNA, and this is the region which gives us this XYZ long non-coding RNA. Okay, so once this XYZ long non-coding RNA, this is transcribed from this region and it goes and binds to the same DNA strand. So like it binds, so this XYZ, this goes and binds to the same DNA strand. We say that it works in cis fashion. Like it is working, it is regulating the DNA of its origin. Okay, so that is what we call the cis fashion. And if it goes and binds to other DNA strands, so let's assume that this is other DNA molecule, and now this XYZ can work in, it can bind to this DNA, to the DNA of its origin as well as to the DNA strand which is not like from which it is not originated. For example, this is the DNA molecule, this is the DNA strand which doesn't have this sequence which codes for XYZ protein. So like when it works, so in cis means it can work on the DNA strand from which it is synthesized, and in trans means it can work on the DNA no matter whether it is the DNA of origin or not. I hope it's clear. So the XIST long non-coding RNA that works in cis fashion means XIST long non-coding RNA. It is transcribed from X chromosome and works on the same chromosome. Okay. So on the contrary, there's other long non-coding RNA known as hot air. It works in trans fashion. I hope you remember when we were learning about the long non-coding RNA, we learned that the hot air, this long non-coding RNA, is involved in the Hox gene silencing. So this can work in trans fashion. Means this long non-coding RNA, this is transcribed from Hoxi locus and it works on Hoxi locus. It works on Hoxi locus. Okay, so the locus from which it is synthesized and locus on which it works, they are different. That is why hot air, this long non-coding RNA, this works in trans fashion, while the XIST long non-coding RNA, this works in cis fashion. Okay, so this was the first point which is important to note. Then another point that exists long non-coding about exists long non-coding RNA is that this exists lncRNA. This has repeat regions which are quite important, and out of those repeat regions there's a repeat region known as rep A or repeat A region, to which this is very crucial because to this region of the XIST transcript, the PRC2 complex, this binds to that. And I hope you remember that when we were learning about the methylation part, histone methylation, we learned that the PRC2 complex, this is very important. This is a silencing complex. This is involved in the gene silencing. Okay, so if you do not remember, let me revise it quick. So this is a DNA. Okay, this is the X chromosome. Let's assume this is X chromosome. And from this same chromosome, the XIST long non-coding RNA is synthesized. So this is XIST long non-coding RNA. Wait a second. So this, uh, so let's assume that this is the XIST long non-coding RNA. This is XIST. Okay, and this is synthesized from this strand itself. So this has a repeat A region. Okay, this exists. Now let's assume that this is the repeat A region. So this pink one is the repeat A region. Okay, so this is repeat A region. Okay, so to this repeat A region, the EZH2, this is the subunit of PRC2 complex, and this is the histone methyl transferase. This is the HKMT, histone lysine methyl transferase. So this does the H3K27 methylation. Okay, which is the again, which it is also related to heterochromatinization. The H3K9 methylation mark is hallmark of heterochromatinization. I hope you remember that. Then we also learned about H3K27 methylation mark, which is also a repressive mark. It is related in the gene silencing because this mark is established by EZH2, by this enzyme, and this enzyme is the subunit of the PRC2 complex. So this is subunit of polycomb repressive complex. So to this rep A region, this is the EZH2. This binds, and this is a part of the PRC2 complex. Okay, so this complex, this scaffold is formed on the X chromosome, and this results into the heterochromatinization. Okay, and this is how X chromosome inactivation is initiated. So for once we initiate the X chromosome inactivation, it spreads on all over the X chromosome. Why? Because very first thing is that the XIST long non-coding RNA, this works as a guide. Okay, we learned about the mechanisms by which long non-coding RNA works, the guide mechanism, scaffold, decoy mechanism. So we learned that in guide mechanism, the long non-coding RNA acts as a guide. It guides it guides the regulatory molecules, the regulatory factors. So in this case, it is PRC2. Okay, or it guides the chromatin remodelers or the factors which are going to change the chromatin environment to the site of the site of the action. So here exists acts as a guide, and it guides the PRC2 complex on the X chromosome, which is going to be inactivated. So it works in cis fashion, and that is why it works on this same X chromosome from which it is transcribed. Okay, so like let's do a quick recap of how the X chromosome inactivation is done. First, we do the counting. So in the counting, we determine the X chromosome to autosome ratio, and in that we decide whether the X chromosome needs to be inactivated or not. So now it is established that yeah, this is female, it has two X chromosomes, so now it needs the X chromosome inactivation. Why? Because the production of RNF12, it reaches to the threshold level. So now once it reaches to the threshold level, the XIST long non-coding RNA gets activated. So once it gets activated, the T6 will be silenced, okay, and XIST will be produced. So once XIST long non-coding is RNA is produced, then comes the choice part. Whether the choice will be random or whether the choice will be skewed, that will be determined. Whether the X chromosome inactivation will be random or it will be imprinted, that will be seen. Okay, the random X chromosome inactivation is a widespread phenomena. Well, imprinted X chromosome inactivation is rare. Okay, it is only seen in the extraembryonic tissue in the mammals, like the placenta or the trophectoderm. Trophectoderm develops into the placenta. So in the once the choice is made, once we like now we have established that the X chromosome inactivation will be random, that exists long non-coding RNA, which is activated due to the threshold level of RNF12. Now it acts in cis fashion, so it inactivates, it goes and binds to the repeat, so it has the repeat A region, okay, and to which the PRC2 complex binds, and now this XIST RNA binds to the X chromosome. Okay, so here you can see in this diagram this XIST RNA to which the PRC2 complexes bind, it binds to the X chromosome. So now this XIST RNA is kind of acting like a guide for the PRC2 complex to where, so PRC2 complex is the chromatin remodeling complex here. So it is going to do the, it is going to change the chromatin architecture. So it is the long non-coding RNA exists is acting as a guide for the PRC2 complex. So PRC2 complex, we know that it has EZH2, which is going to do the methylation of histone. Okay, so this is how XIST goes, binds to the X chromosome from which it is transcribed. So once XIST goes and binds to that X chromosome, now the X chromosome inactivation is initiated. But for the establishment of X chromosome inactivation, this there's a spreading of X chromosome inactivation. Means XIST goes and it wraps around that X chromosome. So like one XIST molecule can recruit other XIST molecule as well. So exist one XIST molecule recruits other XIST molecule, and this is how the whole X chromosome it is wrapped up by the XIST long non-coding RNA. So this is X chromosome, and now it is wrapped up by the XIST long non-coding RNA, and that is going to that is going to result into the X chromosome inactivation. Okay, so this is how once it is initiated, okay, the establishment is done. Establishment of X chromosome inactivation is due to the PRC2 complex, which do the gene silencing. So it activates all of the repressive mark. Okay, it establishes all of the repressive mark, and then there is spreading. So the spreading is because all of the XIST long non-coding RNA, it masks the X chromosome. Okay, so one XIST molecule can recruit other XIST molecule, and this is how the whole X chromosome inactivation is spread on all over the X chromosome, as well as the XIST long non-coding RNA. It is going to mask the X chromosome. So this is how the spreading is done. Once the X chromosome inactivation is spread on all over the X chromosome, sorry, the XIST long non-coding RNA is masking all of the X chromosome. It does some does the two things. First, it adds repressive marks. All of the repressive marks are added due to the crosstalk between the repressive marks. Okay, for like the PRC2 complex does the H3K27 methylation that will lead to H3K9 methylation, which is also a hallmark of heterochromatinization, because this is going to recruit HP1 protein. We have learned about all this in the histone methylation part. And the other thing that XIST long non-coding RNA masking does is that removal of active marks. Active marks, they are removed, while repressive mark, they are established on the X chromosome. So like all of the active marks like H3 acetylation, H4 acetylation, okay, H3K4 methylation, which is also an active mark. All of those active marks are removed so that the chromatin can come into the closed conformation from the open conformation. Okay, so the conformation of chromatin is changed, and all of the repressive marks such as H3K27 methylation, H3K9 methylation, H3K119 ubiquitination. I hope you remember this is also an repressive mark. All of those repressive marks are established. So this is how the spreading of the XIST long non-coding RNA does the X chromosome inactivation. So once this is the X chromosome inactivation so is established and it is spread on all over the X chromosome, The next step is the fourth step, the last step is the maintenance of X chromosome inactivation, and it is very important step. Because once you have established the X chromosome inactivation, you need to maintain that as well, because it need to be maintained throughout the life of the cell, as well as it is maintained in the progeny of the cells. So maintenance of X chromosome is also crucial, and the mechanism of maintenance of X chromosome, it's different if the inactivation is random, and it is different if the inactivation is imprinted. So the mechanisms for the maintenance is different, but the overall maintenance of the X chromosome inactivation is driven by the DNA methylation. Okay, so I hope with this the X chromosome inactivation is clear to you all. If you have any queries, please comment them below. From the next lecture onwards, we are going to learn about genomic imprinting.